When constructing new functionality in cells, your initial design will most likely result in a non-optimal composition. Libraries employ focused diversity to explore the sequence neighborhood around your initial design to identify improved variants. Suppose you want to introduce a biosynthetic pathway into an engineered cell. If all the enzymes express, the substrate chemicals are all available, and there aren't reactions diverting your intermediates to other products, then most likely your cell will produce the target chemical. You might try knocking out some genes in the genome that you think will increase yield, and you might try different regulatory sequences to get the expression level of each enzyme just right, but often is the case that these rational approaches to optimizing the cell only address some problems. If you have difficulty with the expression or folding of a protein, or the protein has poor activity towards the reaction you need it to do, you'll have to make changes internal to that protein sequence. For the scenario in which a nascent activity exists and you just need to improve it, directed evolution techniques are incredibly useful. Directed evolution involves making iterative changes to a sequence to improve its function. Though its use is not restricted to optimizing protein coding sequences, most applications of directed evolution involve optimizing protein sequences. Directed evolution approaches are only one technique useful in protein engineering. One approach is rational design, meaning a person looks at a crystal structure, multiple sequence alignments, and mutagenesis data, and forms hypotheses in their head about modifications to the protein that will improve whatever property they need to optimize. They then design a few genetic constructs and test their hypothesis in the lab. There are also computational tools to do this sort of logic in silico. The most popular of these tools is Rosetta, which is excellent for predicting improvements to protein folding and is making headway into many other functional aspects of protein sequence. There is also an area of protein engineering about modular design in which you fuse coding sequences together either to create new function or improve the folding stability or localization of your protein. The focus of this series of lectures are the library strategies, directed evolution, and combinatorial methods. The term directed evolution is often applied to a broader swath of experiments that includes combinatorial methods, but I will draw a distinction between the two of these in our lectures. Our focus for now is the more narrowly defined directed evolution involving iterative changes to a sequence that each produce an incremental improvement of some aspect of its function.